Welcome everyone to this bioinformatics webinar series. I will introduce today's topic, Streamlined chromosome scale phasing with HiFi sequencing. As you know, long and accurate sequencing reads such as HiFi read give better assemblies. Long reads can uniquely span repeat sequences in a genome, giving a more contiguous assembly graph, whereas short reads cannot span larger repeat sequences, resulting in an entangled assembly graph that leads to fragmentation and sequencing gap. These missing sequences in turn lead to missed genes and limits biological interpretation. Compared to short reads, long reads give a more comprehensive, structural, and functional organizational view of the genome. But length is not the only important factor. As I said, HiFi reads are long and accurate, and PacBio's HiFi read technology reads both strands of a DNA molecule multiple times. The error profile is non-systematic and random, allowing to polish out error and determine a consensus sequence for each DNA molecule. The mean accuracy of this consensus, a HiFi read, is 99.9% or only one error in 1,000 sequence bases. HiFi reads are extraordinarily accurate. They are today the most accurate long reads that are available. They give you an even coverage, so for instance, no dropouts over repetitive DNA sequences. They give you a complete genome, for instance, less gaps in genome assemblies. And the high accuracy gives a low resolution, and read length contributes to long range feature. High accuracy and read length combined allow for excellent variant detection, from small single nucleotide variants to large structural variants. And indeed, the graph on the right shows the results for the Precision FDA challenge, where we show a best performance in class for all variant detection, where a benchmark genome with a known set of truth variants is compared between technologies. And what you see is that HiFi read gives the lowest number of variant call errors compared to other technologies. So why do you need HiFi reads for assemblies? Well, as I said, the long reads span short repeats, and the accuracy allows for differentiation between copies of larger repeats due to polymorphisms. In addition, the accuracy helps to phase haplotypes in diploids and even polyploids, so you no longer end up with a collapsed assembly, but can get a truly haplotype-resolved assemblies. HiFi reads can be applied to small and really big genomes. For instance, the Redwood genome, which has a 54 gigabase DNA content. The HiFi assembly of this genome was more complete than noisy long reads, basically. So long and accurate is what you need for HiFi assemblies. Assemblies with HiFi reads give you great contiguity, high contiguities. They give you a complete genome, minimizing missing bases and fragmented genes. They give you a correct genome, high base accuracy, and phasing of alleles. And the compute is very modest because you virtually have no read error correction to do, giving you smaller file sizes and faster analysis time. So these are the benefits. Now let's discuss some recent results highlighting what those benefits can do in real life. As I'm sure you know, the original human genome wasn't actually complete, and the T2T consortium recently presented the first complete telomere-to-telomere haploid human genome. After years of using a variety of sequence technologies, the T2T consortium switched to the long and reads and high accuracy of HiFi sequencing to resolve this problem. And using this approach, they were able to generate a high-resolution string graph directly from the HiFi reads. And this string graph formed the foundation of the final telomere-to-telomere assembly. As said, the T2T consortium made a complete assembly of a haploid genome, and since people have moved on to more complex assemblies. For instance, the Human Pan Reference Genome Consortium compared methods for the most complete, accurate, haplotype-resolved, and cost-effective diploid genome assemblies that they could found. In their study, HiFi reads gave the best results. Here are some landmarks of the HiFi data listed on the right. HiFi data gave them the largest haplotype phase blocks, lowest haplotype switch errors, highest number of phase bases, and more accurate variant calls across all variant types in challenging regions of the genome. The consortium also generated the first high-quality diploid reference with only 4 gaps on average per chromosome. This allowed them to evaluate haplotype-resolved assembly on a genome-wide scale, including centromeres. And as you can see, today's speaker, Dr. Garg, is one of the co-authors on this publication. Next up is pushing on from single haplotype-resolved genomes to pan-genomes. A pan-genome identifies which portion of the genome are unique and which overlaps between individuals. From an evolutionary perspective, the core genes identified in the pan-genome are likely conserved for function, and there they tend to be conserved within species. In contrast, dispensable genes contribute to the diversity of species, enabling it to adapt to conditions. Thus, the dispensable genes are likely to be evolving faster. As a result, the dispensable genes are a contributor to phenotypic variation. And again, the Human Pan Reference Genome Consortium wants to generate at least 350 haplotype-resolved human genome assemblies. These 700 haplotypes will represent all the majority ethnicities around the globe and will give the full spectrum of genetic variation in the human species. The full first year's effort of more than 40 human genomes was made publicly available last year. All these genomes are based on haplotype-resolved assemblies from HiFi read using HiFi ASM as assembler and were supplemented with other technologies for scaffold. So clearly, I explained to you the benefits of HiFi reads for assemblies and what you can do with those assemblies. But of course, our speaker of today can tell you a bit more about that. Today's presenter is, as said, Dr. Shilpa Garg. She's a scientist working at the interface of biology, computer science, applying to human genetics and precision medicine. She's currently an associate professor at the Center of Biosustainability of the Danish Technical University. Before that, she was an assistant professor at the University of Copenhagen and a postdoc at Harvard Medical School. She has published several high-impact papers in the field of human genetics and cancer research, for instance, the publication that I mentioned earlier and the two that are listed here. Clearly, Dr. Garg is an expert on haplotype-resolved assemblies, and I'm looking forward to her presentation. For now, I thank you for your attention. I will be addressing questions together with Dr. Garg after her presentation. In addition, you can contact PacBio by writing an email to support@pacbio.com. Please enjoy Dr. Garg's presentation.
Hello, everyone. Thanks for joining my talk today, and thanks PacBio for having me. So the topic of my talk is chromosome-scale haplotype-resolved genomics. So first, what is chromosome-scale haplotype-resolved? Imagine, for example, we know humans and other organisms have different number of chromosomes, and each chromosome have a homolog copy. And advances in sequencing technology, especially the HiFi sequencing, where you can get long, accurate reads, has created enormous opportunities to produce the complete genomes right from the sequencing data. Now the goal is, how can we combine HiFi with other technologies to get the full end-to-end haplotype resolved genomes? So little background on the HiFi technology. So the first requirement is you need a high molecular weight DNA. So you start with this high molecular weight DNA, and then you ligate the adapters, and which leads the primers to be annealed and cause this DNA polymerase to be binded. And in this stage, DNA is now circularized. So you see a circularized DNA, and you perform repeated passes of sequences, and finally on the subreads that you get, you call a consensus on every base, and that produces highly accurate reads with the read lengths of more than 25 kilobase pairs on an average in length, and with highly accurate, more than 99.9% accuracy. So we also leverage additional technology, which is Hi-C technology. It's essentially a chromosome capture. It makes use of important spatial information. As we know, these haplotypes form territories in the nuclei. And then, although on the linear scale, they can be tens of megabases apart, but on the spatial location, they are quite close by. So in interest of time, without going detail of the technology, the main take-home message is actually in the right-hand side plot. On the x-axis, you see the genomic loci, and on the y-axis is the contact frequencies. So darker the color is, higher is the contact frequencies between two loci over the genome. So if you look more closely in this map, in about 10 megabases, you see a dark red color suggesting that HiC gives a clear signal in about 10 megabases, but whereas if you go further apart on the genome, it may get pretty low. So now the question is, how can we leverage the advances in the sequencing technologies to produce complete genomes while being at pace in a computationally efficient manner? So a little bit background on the genome assembly field. As we know, this is not a new problem. It started with the Human Genome Project about 2.5 decades ago, where we had short reads about 250 base pairs in length, or Sanger sequencing about 8 kilobase pairs in length. At that time, the reads were not long enough to span these repetitive regions on the genome. As we know, human genome is highly repetitive, with more than 50% is repetitive sequences, either in a tandem fashion or in an interspersed fashion. So the graphs, that there's a beautiful review article from Adam Philippis. So when we produce graphs from the short reads, they look so much messier because we were not able to uniquely place this, find the unique placement of these short reads. Whereas as we move to the long reads, the graph on the right you see are less messier, and you can get better continuous assemblies. But the major limitation in these approaches was the diploid nature of humans or other organisms has been collapsed to make one consensus sequence, whereas we actually want to study the true nature of diploid nature of organisms. So there have been some advances in this area, especially Falcon Unzip and Trio Canu and Trio HiFiism, but there have been some limitations in these approaches. Either they were not able to produce chromosome scale assemblies, or they made use of the trio information, where again limit some of the applications. So to address these limitations, here I proposed a new workflow, which is how can we efficiently combine HiFi and HiC to produce chromosome scale haplotype-resolved assemblies. So as an input, we take first HiFi data, and then we produce continuous sequences, unphased sequences, and then we scaffold these unphased sequences using HiFi to produce even longer sequences, and those are now on the chromosome level. And then in this C step, we map the HiFi reads to these scaffolds and call the structure SNPs. And without using any short reads, we can reliably call SNP mutations directly from the HiFi reads. And we can do this in a very efficient manner in about a few hours of time. And then we use these phase SNPs basically to find the partition of the reads to different haplotypes and assemble these partitions separately to get these phase sequences. And we were interested to see the runtime of this workflow. So we, for the first time, can produce haplotype-resolved assemblies within a day, which previously used to take several weeks of time. And then we benchmarked our approach for widely used genome at C002 and used standard evaluation metrics. So we compared against Trio Canu, and here we observed that we can produce about 3 gigabases of each haplotype, which is as expected in humans. And we also can produce highly continuous genomes, about 25 megabases, which is larger than Trio Canu. And from here, you observe that we can phase about 8 times more than the Trio Canu approach. And then we also perform experiments for other genomes like NA12878 and PGP1, and we observe the similar trend. So this analysis shows us that we are now in a lovely era, that we for the first time could produce chromosome scale haplotype-resolved assemblies in an efficient manner by using HiFi and HiC. And next, we we were also interested to see how can we take these advances to disease diagnostic steps. So we got excited about cancer genomics. As we know, cancer genomes are highly complex and heterogeneous. So essentially, there's a mutation that starts at one cell and then proliferates to millions of cells in quite rapidly, and there is a all level of heterogeneity. For example, intertumor, there are different types of tissues that get cancerous cells, and also the same tumor across multiple geographical locations, there can be a huge heterogeneity. So to understand the heterogeneity, the next question we ask is how can we get a precise landscape, genetic landscape of these cancer genomes? So to answer that, we considered colo 829, which is a melanoma cancer cell line, and we sequenced this using HiFi and HiC to about 30 to 40x coverage, both matched tumor and normal. And then to analyze this complex genomes and complex data types, we actually proposed a new algorithm, which is to use the phasing step right in the overlaps. So we first find the overlaps between in the HiFi reads and also call these SNPs within the overlaps. And then the character within the SNPs, if it is same, then that read belongs to the same haplotype, and otherwise it's belong to a different haplotype. So using these KMER techniques, we have actually now encode this information in the form of a graph, and where in this graph each node is a read and the connections are now the haplotype specific overlaps. And once you encode this information, you start to see these beautiful structures that represents the structure variation complexity that may underlie in these genomes. Especially we see these bubbles which have a common start and end point, and they have these allele paths that can encode all the structure variation. And our next step was how can we bring HiC data on this HiFi unitic space. So we construct this graph, HiFi unitic graph, using this haplotype aware approach. And the next step is how can we map HiC reads in a haplotype aware manner. So the way we do is we find the distinctive KMERs between these alleles in these bubbles, and we use these distinctive KMERs as the anchors to map the HiC reads onto this graph space based on KMERs. And since we are using KMERs, so we are not doing any base level alignment at this stage. So we can do it in a very efficient manner, for example, in a few hours time, compared to the standard traditional approaches that takes about more than a day for doing this step. And then essentially, once we have the HiC read support through this graph, we then use the maximum likelihood models to find the haplotype paths, which are essentially the phased contigs for every component. And we also scaffold these phased contigs based on the neighborhood property to produce the phase scaffolds. So the details are in the paper. Next, we used these new genomes, which are purely in a de novo manner, to call the structure variations. And in this colo 829, we found about 26,000 somatic plus germline structure variations, out of which there are about 13,000 insertions, 9,000 deletions, and 29 duplications. And we also looked into the length distributions of these structure variations. So these SVs can range about 50 base pairs to more than 10,000 base pairs. And you see that in all SV classes, we found different lengths of SVs. So in this way, we can get now a precise and more accurate identification of break finds of structure variations using these high resolution techniques. And My favorite plot is actually this one, in which you, on the x-axis, you see the repeat elements. So you have alus, lines, signs, and simple repeats, and even more complex ones. And on the y-axis, you see the agreement of our call set with the benchmarking competing methods. So in the blue bar represents where our calls are consistent with the single cell based calls, which is more than 19%, which is pretty good. But there are some calls that are represented in the green, which are found in the single cell but are missed in our call set. These are primarily due to the clones that are quite rare, and now in the bulk sequencing we were not able to capture some of the rare clone structure variations happening in the rare clones. But then there are 5% of the calls where we are consistent with the genome with the Netherlands group that produce call set using long erroneous data, and other we are consistent with the New York Genome Center. So this analysis suggests that we can now produce a precise structure variation landscape, even in the repeat elements that was traditionally missed by many approaches. And we also found a p10 deletion, which is about 12 kilobases, and which has already known associations with melanoma cancer cell. So we further looked into few more clinically relevant regions. For example, HLA. As we know, HLA is quite unique and diverse, and it's quite heterogeneous, and it's known to be associations with many diseases like schizophrenia or autoimmune diseases. So what we did, we took our assemblies and we mapped these assemblies to the reference genome, and the color represents the divergence level in the assemblies to the reference. So how darker the color is, higher is the divergence. So here, if we look closely in HLA-AA, we see about like 8 to 9% of divergence to the reference genome. And if we do purely mapping based, or we do erroneous long read based, then we miss these high quality, highly divergent structure. And more importantly, if we look closely in these two haplotypes in this region, so there is a variation in one of the haplotype, not the other. So to capture the variation, importantly, in one of the haplotypes, which may have associations to a specific phenotype in a disease, can be reliably done if we can now reconstruct the full high quality genomes. And then there are few more complex genes like within the HLA, DRB1, DQB1, which are quite rapid, having tandem repeats, and then we can reconstruct these genomes because of the HiFi reads and new methods that can analyze these data sets properly. Next, we looked at another important region, which is KIR, which has also clinical significance. And here, if we look closely, that we observe a divergence of 10% of haplotypes against the reference genome. And this region also has some tandem repeats. So we are in DL3, DL1, and DL2. So we are reliably able to reconstruct these region, KIR region. The next is how can we, so now, like, hope you are convinced we can reconstruct high quality genomes, but how can we take these advances to study genetic diversity and evolution? So for this, we proposed a perspective with my postdocs, chromosome-scale haplotype-resolved genomics. So essentially, you have the multiple genomes of reference quality from multiple populations, and then how can we put together these across populations in a common data structure to study the comparisons well. So here, essentially, the idea is wherever there is the same sequence against multiple genomes, that forms one node, while the differences, they form the alleles in these bubbles. So these bubbles now can encode small variants. It can encode tandem repeats. For example, one sample may carry 3 copies of, whereas the other sample may carry just 1 copy. So all this variation can be easily encoded in the form of a graph structure like this. So pangenomics also enables us to do SV discovery and to study their associated functional role. So uh, beyond humans, pangenomics also has a key role in sustainable agriculture production to find new breeding techniques. Essentially, for example, there are multiple haplotypes. You can find superior haplotypes by different breeding of haplotypes that are associated with improved stress tolerances. So so this is the motivation of pangenomics, but traditionally the pangenomes are essentially the collapsed sequences, a set of sequences, and also they contain many gaps. But more traditionally, a new graph-based methods have been proposed. So one is reference-based. In reference, like you essentially have a reference, a coordinate system from left to right, and then you take the reads from the samples and incorporate the variants from the reads one by one going from left to right. So this approach is simpler because you just have a linear space to work on, whereas it has limitations that there is a huge reference bias. And to overcome this reference bias, there have been new approaches that have been proposed recently, like K-Merkes and WFA, but these approaches have some limitations that it cannot work for a cross-species analysis. So to overcome that, we propose an hierarchical approach where you are given this phylogeny tree. For example, you have classes, genus, and species. And at every level, for example, at the species level, genus level, and class level, you find the representative haplotypes and the representative variation. And for this, at each level, you construct a directed acyclic graph where you have a node, and then you have the edges that represents the adjacency between these node sequences, and you get a DAG for representative sequences at different class level, genus level, and species level. But then we connect these levels by these interlevel connections based on the similar sequences or based on the reference sequencing as per the need of the users. So the advantage of this approach is it is quite flexible and efficient for evolutionary biologists to navigate different levels at multiple resolutions. But the disadvantage is in case you have a new species which is highly divergent, for example, 99% divergence from the given set, at this stage, we don't incorporate this information in the graph. So this is a work in progress. So to conclude, in this talk, I've presented a new graph-based approach that provides a compact representation to combine HiFi and Hi-C data, and how can we leverage these graph methods to produce chromosome-scale haplotype-resolved assemblies in human genomes and pangenomics, and also have demonstrated the importance of these high-quality assemblies, specifically using HiFi to various medical and evolutionary genomics. So as a nutshell, like we are excited to develop new algorithms and data approaches that are highly diverse, highly complex, and we are interested to develop new graph-based methods to yield meaningful insights to enable precision medicine. And I thank PacBio for the support, and thanks mentors and funding agencies, and thanks everyone for your attention.